Gametogenesis is a process by which the bipotential primordial germ cells produce mature gametes. They are known by different names in males and females. In males it is called spermatogenesis and in females it is known as oogenesis. There are certain similarities between the two, like they both involve a meiotic division and the end result of both is going to be a haploid gamete. This process of gametogenesis occurs through four different legs. First, establishment of germ cell population. Second, gametocytogenesis. Third, meiosis. And fourth, metamorphosis. Now, the fourth phase, metamorphosis, is seen in males only. The first leg, establishment of germ cell population. The primordial germ cells, they are derived from epiblast during the second week of intra-uterine life. From then, they migrate in third week and eventually reach the yolk sac. They can be identified by their pale cytoplasm and ovoid shape, and they are rich in alkaline phosphatase. During fourth to sixth week of development, they reach the region of embryo that forms the future gonad, that is somewhere around 10th thoracic segment. Their mitosis results in increase in germ cell population, and their arrival at the indifferent gonad actually induces the formation of gonads. Now there are some cells, primordial germ cells, which do not make it to the site of gonad, and they are the ones thought to give rise to those embryonic tumors known as teratomas. This phase is common for both males and females. Now let us talk about the males. Spermatogenesis. The spermatogenesis is a process by which primordial germ cells give rise to mature sperms. Now these primordial cells, which have inhabited the testis, they lay dormant till puberty. At the time of puberty, due to availability of increased levels of testosterone secreted by interstitial cells of the lytic, spermatogenesis kicks in. In addition to the commencement of spermatogenesis, the other effects of the increased levels of testosterone have they cause growth of the testes, they cause maturation of the seminiferous tubules, and they bring about a lot of secondary sexual characters. Under the influence of this testosterone, the primordial germ cells in a male differentiate into spermatogonia type A. Further, the type A spermatogonia divide mitotically to form type A and type B spermatogonia. Out of the two, the type A spermatogonium stays back as a reserve cell population, whereas type B spermatogonium marches further towards the formation of mature gametes. The type B spermatogonium enlarges and undergoes mitosis to form primary spermatocytes. These primary spermatocytes prepare themselves for the ensuing meiosis I, and their division to form secondary spermatocytes happens via meiosis I, in which chromosome numbers are halved. Each secondary spermatocyte then divides by second meiotic division to form two spermatids. In this division, the DNA content is halved. Each spermatid undergoes spermiogenesis to form a spermatozoon. This spermiogenesis essentially is a process of metamorphosis by which the spermatids transform into the spermatozoon. At the beginning of this spermiogenesis, the spermatid is circular and contains a nucleus, a Golgi apparatus, centriole, mitochondria, and other organelles wrapped in a cytoplasm surrounded by a cell membrane. The spermiogenesis involves formation of acrosome, condensation of the nucleus, formation of neck, middle, and tail pieces, and shedding off of the rest of the cytoplasm. So during this process of spermiogenesis, which is also called as spermatolysis, the head is formed by condensation of the nucleus. The acrosomal cap of the sperm is formed by Golgi apparatus. Proximal centriole and the other centriole, the distal one, gives rise to the axial filament and annulus, and mitochondrial sheath forms the mitochondria around the middle piece. This entire process of spermatogenesis takes about 64 days to complete, and the sperm then enters the lumen of the seminiferous. For a newly formed sperm to be able to fertilize the ovum, it still needs to undergo two processes. The first one is a spermiation, the second one is the capacitation. Spermiation is a process by which the newly formed sperms break free from the Sertoli cells. After breaking off from the Sertoli cells, they undergo functional maturation in the male genital tract, notably in the region of epididymis, which is a temporary storehouse for the sperms. The second process is capacitation, in which there are certain functional changes happening in the acrosome, and this particular process of capacitation, it happens in the female reproductive tract after the sperm has been in the reproductive tract of the female after ejaculation for several hours. Fertilization promoting peptides have a role to play in this. So, to summarize, the primordial germ cells which inhabit the indifferent gonad and induces its development into a testis will eventually form spermatogonium type A. The spermatogonium type A divides mitotically to form a type A and a type B spermatogonium. Spermatogonium type B marches on. It forms the primary spermatocyte by mitotic division, primary spermatocyte by primary spermatocyte undergoes the first meiotic division to give rise to secondary spermatocyte, and secondary spermatocyte by the second meiotic division gives rise to spermatids, and these spermatids transform themselves into sperms by a process of spermiogenesis. A few disorders of spermatogenesis that can be labeled as azoospermia, where no sperm cells are produced in the ejaculate; oligospermia, where the sperm count is low; asthenozoospermia, where the sperm motility is reduced; teratozoospermia is reduced percentage of sperm with normal morphology. Oligoasthenoteratozoospermia is a combination of the above mentioned disorders. Hypospermatogenesis is decreasing the number of the germ cells, that is spermatogonia and the primary spermatids, and there can be a maturation arrest at any phase of the sperm production. The oogenesis or ovogenesis is a process of formation of mature oocytes from the primordial germ cell, that is oogonium. It happens in the female reproductive system, begins before birth, that is in the intrauterine life, somewhere around 6th week of it, and is completed after puberty. This process goes on till the age of menopause. So what happens in oogenesis during the prenatal life? The prenatal maturation of oocytes, primordial germ cells differentiate into oogonia. This occurs in cortex of the ovary. The oogonia divide mitotically to form a cluster of cells surrounded by flat epithelial cells which we call as follicular cells. Many such follicles are formed, and you can see that formation going on till the end of third month of intrauterine life. The oogonia continue to divide by mitosis, and some of them enlarge and form primary oocytes. But this further maturation is arrested at the level of prophase 1 of the primary oocyte, and the arrest is happening because of a certain factor called oocyte maturation inhibitor, which is secreted by the follicular cell. From then on till birth, the number of oocytes reduces, and in the postnatal maturation, the number of surviving primary oocytes remain in prophase 1 arrested, of course, till puberty, and each oocyte is surrounded by a layer of flattened follicular epithelial cells. Such an assembly in which the center is occupied by a primary oocyte and surrounded by follicular epithelial cells, such an assembly is known as a primordial follicle. Now, at the onset of puberty, due to availability of the gonadotropins, under the influence of the gonadotropins, about 5 to 15 primordial follicles start maturing in every ovarian cycle. The primary oocytes increase in size, follicular cells become cuboidal, and then they become columnar. So, change in the character of the follicular cell is marked as a sign of oocyte activation or follicular activation. So, a primordial follicle gets to form a primary follicle in which the cell character of the surrounding changes from squamous to cuboidal. From cuboidal, it goes on to increase in the cell numbers, that is called a multilamellar primary follicle. Then the cell divide so rapidly that eventually you get to see some spaces. The spaces soon coalesce and they give rise to a large cavity called the antrum. Such a follicle is called the antral follicle. The antrum is filled with tissue fluid secreted from the surrounding cells. The tissue fluid is called liquor folliculi, And gradually, this follicle, secondary follicle or antral follicle, keeps enlarging as more and more fluid seeps through the intercellular spaces and the cavity inside increases. Eventually, we get a fully formed Graafian follicle or a mature follicle, which is about to rupture at the time of ovulation. Now, at the time of ovulation, due to the LH surge, the wall of the follicle breaks off and the uh, developing ovum surrounded by follicular cells is extruded out into the general peritoneal cavity to be picked up by the fimbriated end of the fallopian tube. The rest of the wall of the follicle, under the influence of LH, luteinizing hormone, proliferates and gives rise to a yellow body called corpus luteum. This corpus luteum has got cells necessary to synthesize progesterone now to bring about secretory changes in the endometrium. If this ovulated developing ovum does not fertilize, then the further development will not happen. The buildup of progesterone will eventually wean off, and that would mark the beginning of next menstrual cycle. It is necessary to note that this meiotic division that got arrested during the usual development will be completed only at the time of fertilization, where the secondary oocyte will form and the secondary oocyte will eventually complete its second meiotic division and become a mature ovum transiently, because immediately after formation of that, the male and the female pronuclei are going to fuse and a zygote will be formed. The usually seen abnormalities in the oogenesis are oocyte maturation arrest that can occur at any stage. It may be due to meiosis defect, which can lead to abnormal chromosome segregation. The other anomaly can be chromosomal abnormalities. These can be either numerical or structural anomalies of the chromosomes. There can be premature ovarian insufficiency that can cause oocyte abnormalities, or there might be mitochondrial abnormalities that can cause oocyte abnormalities again. And a lot of them has got significant bearing with maternal age. The more advanced the maternal age is, more are the chances of uh, finding abnormalities in oogenesis. That is gametogenesis for you. Happy learning.